Now it is my great honor to introduce the moderator and the next session, which is very exciting. We'll discuss a very important question about animal model. The moderator will be Professor Stanley Perlman. He's a professor from University of Iowa. He is also a well-known expert of coronavirus. Actually, he was a panel member last time. He did so well, we decided to invite him back again. So Stanley, that's yours, the panel now.
Okay, thank you. So the title of this session is Challenges for COVID-19 Vaccine Development. And what we're going to focus on here are preclinical animal models. And the first speaker is going to be Bart Haagmans from Erasmus in the Netherlands. Bart trained at Utrecht. His interest for many years has been on emerging virus infections. He's made many contributions to our understanding of the molecular biology of the virus. He helped identify the MERS receptor for the virus. He's developed serological assays, and he's developed vaccines for MERS and many other pathogens as well, both in camels and people. Now he's working on SARS-CoV-2 vaccines. And so take it away, Bart.
Okay, thanks, Stan. So this is to kick off this session on the roles of animal models. So I will give a short summary in a few minutes on the findings actually of the WHO working group on animal models that's quite active. So next slide. So basically, I think to summarize it is that the animal models available are mice, hamsters, non-human primates, and ferrets. What we know at the moment on the mouse models is that given the fact that the murine ACE2 does not effectively bind the viral spike protein of SARS-CoV-2, it's difficult for the virus to enter cells. So you need to adapt either the virus or use human ACE2 transgenically expressed in mice to make these animals susceptible, and that is what's being done currently. Of course, it makes it a bit difficult to use mice, as you need these mice now or the viruses that are adapted. An alternative strategy would be, and that is explored by Stan Perlman, actually, and that's quite elegant, is the transduction of normal mice with an adenovirus that expresses the human ACE2, and that's also a nice model. More on the long term, I think there's the collaborative cross mice that have different genotypes and then also phenotypes which could be used, and of course a combination of human ACE2 transgenic mice with human hematopoiesis. One obvious advantage is that for these mice, tools are available to characterize the immune response in quite detail. So this is of course quite nice, but some of these models you need to adapt the virus, and that would not reflect the virus that are circulating at the moment. Next slide. A very nice model, I think, and used worldwide, and that's basically because it's also commercially available quite easily, because these are just the wild type Syrian hamsters that show upper and lower respiratory tract infection, and they also show some clinical signs. So that can be seen that there is weight loss. It's important to notice that after day 5 they gain weight, and so they recover. But infiltration of the lungs can be seen by cells of the immune system, and there's high level of replication. So I think this is a very nice model that can be used as one of the steps to characterize vaccines also, both for immunogenicity. Although compared to mice, of course, this is a bit more restricted because tools are less developed for hamsters, although there are also knockout hamsters available at the moment. Next slide. So then going to the non-human primates, and this has been shown already earlier, and I think the lead vaccines that are now presented also today used non-human primates quite extensively to show immunogenicity of vaccines. And upon challenge with SARS-CoV-2, actually the non-human primates, it has been shown for cynos, rhesus, but also African greens, that the virus replicates in the upper and the lower respiratory tract. And it would be important to have a bit more severe disease in non-human primates. This has been seen in African green monkeys, but Not so much in our hands when you're using, for example, aged animals, and that's shown in the graph, so that you see shedding for a longer period, but we did not really have evidence for a really severe disease in the lower respiratory tract. But still, I think this is a very useful model, and it has been used for many of the vaccine candidates to date, I think, for the initial testing, especially for mild disease. So tools are available, but no severe clinical disease. Next slide. Other animal models, ferrets. Ferrets actually are quite interesting in that they do show an upper respiratory tract infection. They can be infected with a low dose, and that is also seen when these animals are used for virus transmission. We have evidence also that not only contact animals, as shown on the right, that are in direct contact with an infected animal, but also upon transmission through the air, these animals can transmit the virus to recipients. So this is an interesting model to study transmission. I should say that hamsters also transmit the virus. Both animal models show upper respiratory tract infection, which is quite efficient. In contrast to hamsters, ferrets don't show efficient replication in the lower respiratory tract, which is quite interesting because the SARS-CoV-1 replicates to high titers in the lower respiratory tract. There are some histological pathological changes, but these are quite limited in ferrets. Furthermore, there is evidence for the feline species, cats, susceptible, but the question is how useful this is a model to test vaccine candidates. So overall, I think there's a combination of several animal models, not only small animals, especially the hamster, and on the long term, also the mice that are available to test vaccines for immunogenicity. And as said, I think by Marian Gruber, I think it's important for these animals to check for selection of dose, for example, but also safety. We haven't seen issues like antibody-dependent enhancement in animal models, and also not enhanced respiratory disease. So this calls for the development of these models to see whether positive controls of vaccines other than currently developed would also induce these enhanced disease in these models. So that would be important to check. And last, I think one question would be whether the data provided in animal models, next slide, on the product effectiveness, so whether there's evidence for effectiveness in the animal models, whether in the end, maybe at a later stage in the outbreak, would be needed to get licensing of vaccines in the absence of outbreak, for example. And that is following the animal rule. And I think this is still a matter of discussion, which we can discuss in this session. Of course, also as indicated, I think if there would be a correlate of protection defined, for example, neutralizing antibody titer, that could be also quite informative in getting this further. So with that, I want to close and give back to Stan.
Thank you. I think what we're going to do is go through all the presentations, then have questions at the end. So the next presentation is by Vincent Munster from the Rocky Mountain Labs. He's the Chief of Virus Ecology, and Rocky Mountain Labs are part of the National Institutes of Health. Rocky Mountain Labs has made many contributions to our understanding of emerging infections. They have a BSL-3 and a BSL-4 laboratory. Vincent himself has worked on vaccines, antiviral studies, environmental studies, and virus pathogenesis. He's done field work in Africa, and he's worked on both coronaviruses, filoviruses, and other bad actors. So with that, Vincent, can you take it away?
Thank you, Stan. Can everybody hear me just fine?
Yes.
I assume that's a yes. Next slide, please. Yeah, I was going to talk a little bit about like my personal experiences, and particularly what I think put us in this current situation where I think as like a global infectious disease community / vaccine community are in a relatively good spot. And obviously, after the Ebola virus outbreak in West Africa, I think there was a big realization that on one hand, there were a lot of like vaccines in preclinical development, but it was very hard to move these vaccines in actual clinical trials for a whole number of reasons. And it was really clear that we needed to advance these vaccines further in order to be more prepared. And obviously that was a combination between CEPI, Coalition for Epidemic Preparedness Innovations, in combination with WHO, with the WHO Blueprint Priority Diseases, where there are a number of diseases who were really stratified in being a high potential risk of causing these large-scale outbreaks. We have, for instance, the Middle East Respiratory Syndrome coronavirus, something which both Stanley and Bart worked heavily on, Lassa virus, chikungunya, Nipah, Rift, and then obviously disease X, which I think is a situation we find ourselves in now, which turned out to be the SARS coronavirus too. And I think this is a little bit like preaching to the choir, obviously, but the success of this vaccination is seen in this graph, which was actually constructed by one of my grad students, Jeremiah Madsen, who's working on Ebola. So in orange, we see the human cases during the West African outbreak in 2013 and 2015. So really rapid expansion, topping at 28,000 people infected and over 11,000 people actually succumbed to disease. And this outbreak was quenched by classic countermeasures like isolation. And then in the lower, in purple, we see actually the recent outbreak in Eastern DRC, which actually has the same level of problems with their public health infrastructure and political problems. But that outbreak did not explode. And the reason for that is that we were able to move the VSV Ebola vaccine, partly developed by Heinz Feldman in our lab, into Eastern DRC, and allowed us to actually stop the uncontrolled spread, which what happened in West Africa. Next slide, please. So as Bart already said, typically we have this lineup of how we start doing these kind of investigations in countermeasures, medical countermeasures, whether that would be vaccines or antivirals, starting with preclinical small animal models, preclinical non-human primate models, and then moving effective preclinical development into phase 1, 2, and 3 clinical trials. Next slide, please. So one of the things we actually worked on was a very related beta coronavirus, the Middle East Respiratory Syndrome coronavirus. And that's exactly what we did. We made a vaccine together with Sarah Gilbert at the Jenner Institute against Middle East Respiratory Syndrome coronavirus using the spike. So actually very much the same as all the other vaccine approaches now utilized with SARS-CoV-2. We tested in mice. Next slide, please. We went to monkeys. Next slide, please. And we went into clinical trials, which I think is really for us, it's a blueprint how to relatively rapidly move these preclinical data into clinical trials. The problem is that if we look at the timeline over it, this is 5 years of work, which obviously if you are in the middle of an outbreak, that doesn't really help you that much. Next slide, please. So this is based on some of the data Bart just showed. We developed a non-human primate model. There are different animal models. So at that time, there was no small animal model for challenge. So we used a small animal just to look at the efficacy of the vaccine to induce both a humoral as well as cellular response, but we didn't do a challenge. We set up a rhesus macaque model and we used that in challenge. That study is currently accepted in Nature, but then we rapidly moved this in a combined phase 1 and phase 2 clinical trial, again in the UK by Sarah Gilbert's group, and that was published yesterday. And you can see that this really compresses the timeline of how we can move these studies within 4 or 5 years into 3 or 4 months, which is, I think, quite unprecedented. Next slide, please. So obviously, we're now in a situation where we have multiple vaccine candidates, we have multiple front runners, we have multiple clinical trials. The biggest problem is, of course, that we now need to move in those phase 3 clinical trials where you need to have active circulation in order to really determine efficacy. Obviously, some of these clinical trials are really focused to start in areas with active circulation, for instance, the US, Brazil, and South Africa. And if you go back to the previous one of the previous presentation by Marion Gruber, that for the FDA is really, for this particular pathogen, the way to move forward in going for licensure. So obviously there are alternatives, like one is something we can discuss a little bit later on in this session, is the FDA animal rule, which allows efficacy findings from adequate and well-controlled animal studies to support licensure. The problem is, because we're so early in this outbreak and we don't really have good data on the correlates of protection yet, this might be only really like a strategy for a long-term approval. And then secondly, which has been proposed is chimp studies, utilizing control human infection models, where you can really do challenge studies in humans. So these are different strategies to support, for instance, phase 3 clinical trials. And with that, I'll give it back to Stanley and the next presenter.
Thanks, Vincent. So the third presenter is Linda Saif, who's a distinguished professor at Ohio State University. Linda's been working on veterinary viruses, including coronaviruses, for many years, over 40 years. She's well known for her work on pathogenesis, vaccine development, antiviral studies, and she studies viruses other than coronaviruses, including rotavirus and norovirus. She is also a member of the National Academy of Sciences of the U.S. So, Linda, take it away.
Yeah. Thank you very much, Stan, for the nice introduction. And I'm going to try to fill in a little about challenges in terms of animal models for COVID-19, since we already heard the overview about the various animal models. So next slide, please. So there are quite a few unknowns in terms of the pathogenesis and immunity to SARS-CoV-2 in humans. So this presents challenges for trying to develop an optimal animal model that mimics the situation in humans. So one of the first things is we don't clearly understand the pathogenesis of SARS-CoV-2. Is it really limited to the respiratory tract? And what causes it to, in some occasions, to also apparently infect the gut? And in terms of the first site, the upper respiratory tract infection, how does then the virus disseminate to the lung, the intestine, or other organs? Another big important question is, does severe disease result from the magnitude of infection or immunopathology with a cytokine storm, or both? We certainly don't know the correlates of protective immunity, the role of innate humoral cellular immunity in protection, and a question that has not really been addressed so far, I think, in most all of the clinical studies that I've seen to date, is the role of mucosal immunity to prevent the nasal shedding. We also don't know whether immunity to natural infection can occur after recovery to prevent reinfection, and we don't really know about the duration of immunity. So the first stage vaccines, the critical thing will be to prevent the severe disease, but we would also hope these vaccines would block nasal shedding and transmission. Next slide, please. So there are challenges for animal models and COVID-19 vaccines. One of the major challenges that the animal models may not reflect the vaccine responses in the high-risk groups. And this is especially true for the elderly or those with comorbidities such as diabetes, heart and respiratory disease, obesity. We know that there's a syndrome of inflammation, it's called, and also immunosenescence in the elderly with reduced innate immunity. And this can lead to altered immune responses compared to responses in young, healthy individuals. Another area that's not considered in these models is the impact of medications for chronic conditions may infect disease or immune responses. An example is that the statins affect one's susceptibility to norovirus. So we should try to use aged animals or animal models with the above conditions. There are various animal models available to study diabetes, heart disease, obesity, and so forth. And for vaccines, the message here is that in the elderly, we may need higher dose, like influenza vaccines, better adjuvants, or multiple doses. Another parameter that's important, the vaccines will be probably used in two populations, those who are SARS coronavirus 2 naive versus recovered individuals, and these people will have variable levels of pre-existing immunity. So at current, there's no thought about testing animals that have recovered from COVID-19 with waning immunity in terms of looking at efficacy versus adverse events such as ADE, which may vary. And then another question is, will any adverse events that are seen in animals actually reflect those in humans or vice versa? Next slide, please. And so the last slide just summarizes some general concept in regard to the animal models. So some animal models are inbred, such as mice, and this may not reflect the heterogeneous immune responses in humans or using outbred animal models. So it will be important to test vaccines in outbred animals that reflect the full repertoire of the responses and also potential for adverse effects. We all know the animal models do not reflect the human immune system, even the non-human primates. There may be differences in the correlates of immunity. There may be differences in immunopathology. So one future approach is to reconstitute the immunodeficient mice with human immune cells, as Bart also mentioned. And then an area that hasn't been covered at all, as far as I can see in the literature in regard to COVID-19, we know that the animal models also lack the human microbiota, and that in the elderly, there's more of a pro-inflammatory microbiota. There's likely dysbiosis in the microbiota in COVID-19 severe cases. And now there's many publications that document that the microbiota influence immune responses, the epithelial barrier, and also importantly in the context of COVID-19 inflammation. So in our lab, we've been working on reconstituting germ-free piglets with the human microbiota. We've successfully done that to show that they can essentially be colonized with the human microbiota and maintain the colonized microbiota. And then we've been looking at the impact of this on human rotavirus vaccines. So with that, I'll turn it back over to Stan. Thank you.
Thank you, Linda. So I think we have time for some questions. Let's see if there's some. I don't know if there are. Are there questions here? Shan, do you see any questions for this session?
Yes, there are some, such as the guinea pig or mini pig with metastatic syndrome, metabolic syndrome might demonstrate efficacy against severe disease. What do people think about that? That was one of the questions.
Okay, that's the first one. Okay. So does anyone want to answer that, whether guinea pigs or mini pigs with metabolic syndrome might be useful models for studying the severe disease in humans?
So for mini pigs, I think the idea is that pigs are not susceptible for SARS-CoV-2, so that would be a problem.
Okay.
Guinea pigs would be something different. I don't know of efforts to test guinea pigs at the moment for susceptibility. Over.
Actually, as a follow-up for that, there's a question that I have about rabbits. Do we know if rabbits are susceptible to this virus?
So actually, we did experiments with rabbits, and rabbits are susceptible. That was in relation to also outbreaks here in the Netherlands in mink, and also farms there with other animal species, including rabbits. So there would be a risk assessment to see whether rabbits are susceptible and potentially can transmit the virus. That is less likely, I would say.
And then there's another question, actually just along the same lines, about the role of whether minks would be useful for animal models. They certainly get sick, but I don't know if anyone wants to work with minks.
Yeah, they're planning to kill all minks in the Netherlands, so there are no animals left, so that's a problem.
That would be a problem. Okay, let me see.
There are also people ask about cats. What are your view, any of you, about the cats?
Well, this is Vincent. So I think people have to realize that typically how you would choose your animal model is, on one hand, is like how the disease manifestations are there, but it's also like how many reagents are available and how easy can you actually utilize these models in a high containment setting. So although it might be very useful to look a little bit more in detail into cats and pathogenesis and cats and transmission, I cannot really see that particular model moving into really like a high throughput setup. And I think particularly if you look at countermeasures, medical countermeasures, it's all about your ability to do things high throughput in sufficient numbers to really get robust data in how effective your countermeasures are. And I think in that sense, like all the different mouse models, including like Stanley's K18 mouse model, I think they show a lot of promise where we see more severe disease, including lethality, and that would bridge then nicely into larger non-human primate models.
Yeah, one of the questions along the same lines, is there any information yet about whether cats can spread the infection to humans?
I think basically, as a virologist, if a cat gets infected and it sheds sufficient amount of virus, then obviously it can get transmitted. If there is formal proof about that, I don't know. Do you know, Bart?
I think there's no formal proof at the moment, but I agree with you. This could be a route of transmission potentially.
Okay, and Linda, here's a question for you. It says, how does a different microbiome affect the efficacy of vaccines?
So that's a very broad, open-ended question, I would have to say. In short, we don't completely know the answers to that. It's going to vary depending on the age group, the microbiome, and the vaccine you're testing. What we've been trying to recapitulate in our germ-free pig model with rotavirus is a human infant microbiome from malnourished children to see the impact on vaccines. So I think you would have to look at that in the context of the different microbiota in terms of the regions which are influenced by diet and all of these factors to really try to recapitulate that.
Okay, and then another question here is this. Each one of you can say in one sentence, maybe. What do you each of you think is the best animal model to study COVID-19 now?
I can start with that. It really depends on the question you're asking. If you want to develop like rapid countermeasures and vaccines, combination between small and non-human primates would be best. If you're looking into pathogenesis, you of course need a more severe disease model. So with animal models, it always comes down to what particular question you're trying to address.
Bart or Linda?
I agree. So I would have the same answer. So, but so far, I think the models that are there, so especially hamster and non-human primate, I think suit quite well to test the vaccines on preclinical. So it's up all to the field trials, I think, the clinical trials to show that they're really effective. Over.
I would agree, but I would also urge people to try to develop these models better to reflect the high susceptibility age groups, as I mentioned. I think we could do a better job of that, and then look at some of these variables and parameters that are going to affect the outcome of vaccines in this highly susceptible population.
Okay, so I think this will be the last question. So one person commented that there seems to be little justification for the continuing use of non-human primates for SARS-CoV-2. Could each of you comment on that?
Well, I disagree. I think there's a great need for good NHP studies particularly for countermeasures, but also, as Linda said, better understanding correlates of pathogenesis and underlying conditions. So I disagree. I think there is a big need for non-human primate studies.
Bart?
Yeah, I also disagree. So I think the studies presented, I think already and published on the vaccine candidate tested, I think probably this has to be shown in the follow-up, but I think they show the usefulness of these animal models. And yes, I fully disagree. So.
Okay,
So-
Linda?
So I would disagree too. I think if you look at the literature, you already see there are differences in the young primates, non-human primates versus the older groups. So we could start using that more in terms of the vaccine effects. And also, I think the non-human primates, you may be more likely to see an adverse event that may be more similar to what we might encounter in humans than what we might see in the other animal models. So I think that's critical too.
Okay, thank you. So,
Yeah, and I think a last comment is that, and that's of course it's not really dictated by us in the field, whether that would be Linda or Bart or me. It is dictated by, for instance, FDA or other agencies allowing to move, for instance, certain vaccines into clinical trials. They actually want to see non-human primate data. So if you're not able to show that data, your countermeasure will not be approved to actually move into clinical trials.
okay, thank you. Shan, should we stop now?
Sure. I think that's wonderful. I want to thank Dr. Stanley Perlman for moderating a great discussions, and also the 3 panel speakers. You invite most questions. Unfortunately, I just wish we have more time. Any good party have an ending, so we wish we'll have more time, and next time we'll invite you back again. So I think we are almost done. I want to take this opportunity to thank all the speakers and the chairs for another wonderful Congress on the top of COVID-19 vaccine. Also, as asked by my co-chairs, I want to provide a brief summary of the 2 virtual Congress we have had so far. One we had 1 month ago. So far you can see, I just put slides there. We covered the 8 leading COVID-19 vaccine programs based on 4 different vaccine delivery platforms. I rank them from the traditional older one, like Dr. George Gao present in Activate, used widely in China, to recombinant viral particle, to viral vector, and the viral vector, and then the nucleic acid. You see the hot one is the viral vector and the nucleic acid vaccines. So clearly, this is the modern technology are dominant among the candidate of COVID-19 vaccine. It would be exciting if one of the unexpected good outcome of COVID-19 pandemic is the validation of some of these new vaccine technologies. However, we know at the same time, lack of previous successful experience with such new vaccine technology should remind us the potential challenge of moving this vaccine into marketplace, especially considering the time, demand, and then the doses number needed for the global vaccine application. Therefore, our Vaccine Congress series also included some strategic discussion. We covered 4 topics so far. That is the basic biology and also the global collaboration in the first Congress. And today at the first session, we had 2 topics, the regulatory consideration and the largest scale clinical trial during today's first session. We also discussed about some of the technical tool. For example, the last time we discussed whether it is acceptable to use the live virus to do a human challenge. As you know, after our Congress, actually we see a letter signed by over 100 prominent scientists suggesting to do so. Also, today we discuss animal model. So both of them are going to be potential very useful tool to accelerate the vaccine ultimate licensure and showing the efficacy. In the next Congress, we aim to cover some basic science. Since we already cover basic different technology, we want to understand more back to science to guide us to do more and better vaccine research. We hope our Congress series are informative and useful to global vaccine community, as well as to anyone who would like to know the most updated progress and thinking of COVID-19 vaccine development. At the same time, my co-chairs and me share the same feedback from many attendees. That is, we wish vaccine developers can share more data from their clinical trials. We know today we added some new fresh data, which is very exciting. ISV would like to thank our partners, including CEPI and other regional vaccine societies, such as from Canada, South Korea, and Japan, for their advice and support. We also want to thank sponsors at this difficult time to give us financial support, and their name and logo can be found at our website. The third Congress and the last one of this series will be held on August 25th, also a Tuesday. We will announce the detailed time and program in the coming weeks. For those who missed the first or today's full program, the video recording will be available at YouTube. Please search by International Society for Vaccines. So finally, I want to thank my co-chairs, Linda and Margaret, for their great contribution and teamwork, and the ISV Congress operation team for another successful virtual Congress. Goodbye, everyone, and we're looking forward to seeing you in about 1 month.